Why is Gram staining such an indispensable technique in microbiology? That's a question that has echoed through countless laboratories and classrooms around the globe. Gram staining, named after Danish bacteriologist Hans Christian Gram, is a crucial step in identifying bacteria and understanding their characteristics. This procedure differentiates bacteria into two fundamental groups, gram-positive and gram-negative. This differentiation is based on the chemical and physical properties of their cell walls. Let's dive into the nitty-gritty of the Gram staining process. It's a fascinating journey that unfolds in 4 primary stages. The first step is applying a purple crystal violet stain to a heat-fixed smear of bacterial culture. The heat fixing kills some bacteria but makes sure others stick to the slide. It's a delicate balance. The crystal violet stain penetrates the bacterial cell wall, coloring all cells a uniform shade of purple. Next, we introduce Gram's iodine, which forms a complex with crystal violet and traps it in the cell. This iodine is often referred to as a mordant, a substance that helps the stain bind to the cells. The third step is a bit of a game changer. We wash the smear with a decolorizer such as alcohol or acetone. The solvent seeps into the cell wall, disrupting its structure. The gram-negative bacteria, with their thin cell walls, can't hold on to the crystal violet iodine complex and get decolorized. However, the gram-positive bacteria, boasting thicker cell walls, retain the stain. Finally, we counterstain with a red dye called safranin. This step colors the decolorized gram-negative bacteria red, while the gram-positive bacteria remain purple. The result? A slide that beautifully illustrates the difference between gram-positive and gram-negative bacteria. But what does all this mean? Simply put, the Gram stain method is a fundamental tool in microbiology. It provides a snapshot of the bacterial world, revealing key differences in cell wall structure that impact bacterial behavior, including how they respond to antibiotics. For instance, gram-positive bacteria, like Streptococcus and Staphylococcus, have a thick layer of peptidoglycan in their cell wall that retains the purple crystal violet stain. On the other hand, gram-negative bacteria, such as Escherichia coli and Salmonella, have a thinner peptidoglycan layer and a lipid-rich outer membrane that doesn't hold the stain, but takes up the red counter stain instead. In summary, Gram staining is a simple, yet powerful technique that allows us to classify bacteria based on their cell wall characteristics. It's a world painted in shades of purple and red, a world that, thanks to Hans Christian Gram, we can explore and understand with a few drops of stain and a good microscope.